Very good afternoon, students. How are you people? Namaskar. How is everything in this afternoon? How is your preparation? How are you feeling? How is your health? Okay. So, for the people who have already been tuned with me from last two classes, you know that I am dealing with respiration in plants, chapter 14, class 11. For the students who are getting newly into this, this class, I am Abhinaya. I am going to handle biology. I am one of the biology faculty. So, I will be handling with ETC and also tell you that respiration is what, okay, in terms of metabolism. We raised a curious question in our previous classes saying is whether respiration is catabolic or anabolic or both or something else. So, about that we are going to talk about in this class, okay. So, we were into the cell from past two classes. What do I mean like this? I mean, we were into the cell means what? We talked about glycolysis being inside the cytoplasm. We talked about Krebs cycle being inside the mitochondria. We are still inside the mitochondria because we are going to enter the inner membrane of mitochondria and see how is this ETC cycle. Okay, it is called as electron transport chain. It will be also called as electron transport system. Okay, we will see that how so many reduced coenzymes were been formed in our previous classes, isn't it? We talked about so many reduced coenzymes formed in glycolysis or pyruvate oxidation or link reaction, and what is going to happen with this reduced coenzyme form? They are just floating around in the cell. So, we are going to use this reduced coenzyme, we are going to oxidize them, release electron, push them stepwise into complexes. The protons released on transfer of electrons through this complexes, we are going to push them into ATP synthase and see how ATP synthesis is going to drive away in a topic called as oxidative phosphorylation. Okay. So, as I said, what did we do previous class is we studied about fermentation, we studied about oxidative decarboxylation of pyruvate, that is nothing but link reaction, Krebs cycle, overall output of Krebs cycle. We came up saying that there is 3 molecule of NADH, 1 molecule of FADH, 2 and 1 molecule of ATP. These are all released into the overall Krebs cycle from 1 molecule of pyruvate. We studied about fate of pyruvate, right, the previous class. CPP question most of the students have gone wrong is, from this I can get to know that you have आपको कुछ दिक्कत है I think in understanding link reaction, that's nothing but pyruvate oxidation. Most of them have answered the correct option as acetyl CoA. I'm sorry, most of them have given the answer as B. I'm sorry. So, most of them have said it is citric acid, but it is wrong. The answer is C. Now, how it is actually I am asking the question is, pyruvate enters the Krebs cycle in the form of, I told pyruvate is little unstable to get inside Krebs cycle. Okay, so that is why it gets converted into acetyl CoA. So, this is the stable form of pyruvate which is getting inside Krebs cycle. Acetyl CoA along with oxaloacetic acid, the 4 carbon compound, 3 carbon compound, this is actually forming citric acid. Please note the difference. The first stable compound which is formed in Krebs cycle is citric acid. The compound which is getting inside Krebs cycle, pyruvate enters Krebs cycle in the form of acetyl CoA. Okay, please note this difference. Okay, if you haven't understood, get back to the previous class, read about link reaction or pyruvate oxidation. You will get to know which is that form of pyruvate getting inside Krebs cycle. So, as we started off, हम पढ़ेंगे आज ETS, नहीं तो आप उसको ETC भी बोल सकते हो, electron transport chain or system. We are going to study about oxidative phosphorylation. In my previous class, I have been saying about substrate level phosphorylation. Ab, interestingly, we are going to study about oxidative phosphorylation. What is that? Keep tuned with me. Okay, we'll study as we keep discussing the core concepts of this class. Then we are going to study overall ETS or ETC. There should be some purpose for any or the other process which has been taking place in the cell, right? It should release something which is going to be valuable for the cell. Okay, so this reaction, that is this chain which we are talking, what is it going to put out? That's what we are going to talk, okay, in overall ETC. Then the question, amphibolic pathway, I told what is respiration, we'll talk about that also. Quickly, electron transport system. Now, can you see this? Okay, now this is a chain. You can take a chain of beads also. Now, this is the chain, electron transport chain. This is how it look inside the inner membrane of mitochondria. You guys have all studied about structure of mitochondria, the cell, the unit of life, right? So, look at the diagram I have been pointing at. You have an outer membrane, you have an inner membrane. These two membranes are separated by intermembrane space. Why am I actually talking about this? Where do you think is the location for the system or chain I have been talking about? Is the inner membrane. अगर आप inner membrane पे जाओगे, आपको chain of complexes ऐसे दिखेंगे. वहाँ पर 4 complexes है. This constitutes electron transport chain. यहाँ पर क्या होता है, if you see, is ये जो reduced coenzymes है, everything comes to this chain. They'll start losing their electrons and protons. Electrons more predominantly from top to bottom. That is from complex 1 to 4. There are 4 loops I've kept here. I don't know whether you are able to see this. 4 loops actually. Okay. From one complex to the other complex, the electron keeps going. That's why I've put the question. The first one is, what do we mean by sending down the electrons through this system or chain? Is as we keep sending it down, okay, every transfer it will release protons. So this protons is utilized to drive off ATP synthesis. That's what wholly this electron transport chain is. How is this getting maintained? Okay. Now this electron transport chain, whichever I was talking, it was unidirectional. See, you see the 4 complexes here. It is unidirectional. It goes from the higher redox potential to the lower redox potential. That means the redox potential, that means simultaneous reduction and oxidation keeps taking place in this complex. Maintaining that redox thing is called redox potential. Okay, maintaining redox potential from higher redox potential to lower redox potential, the electrons keep moving. So this is what we said as sending down the electrons by maintaining the redox potential. That's why ETC becomes unidirectional. That means from lower redox potential, from down to the higher complexes, the electrons cannot move. Nicely designed, no? Oh, wait a minute. I told 4 complexes. You must have studied in your textbook or you've come across also. There are 5 complexes, isn't it? So, all the protons which has been released by these complexes, it is actually utilized by the complex 5. I added one more loop. It is not the part of ETC, but it utilizes protons by, sent by these complexes. That is ATP synthase. We'll talk about it much more in detail diagrammatically through our slides. In touch with me. Let's know something else also about ETC, something more a step high, because every student in a class wants to know a step high to become brighter, or to become outstanding in the class. So what is that something that we have to know more about ETC is, what is the input of ETC? Now see, I keep saying that any process you have been studying, glycolysis, Krebs cycle or ETC or anything else in photosynthesis also, think it is like preparing some recipe, okay, like preparing some food. So for food, what we have to see, input, what and all we are going to put in, what is the output which we are going to see, and how is it getting derived off. The same like that, the input for our ETC is going to be NADH from glycolysis, reduced coenzyme, okay. Pyruvate oxidation, we talked in link reaction, 2 molecules of NADH is produced out. Krebs cycle, 3 molecules of NADH, just now I wrote when we talked about what we studied in previous class, okay, and FADH2 from Krebs cycle. FADH2 comes only in Krebs cycle, okay. So these are all the input for our ETC cycle. What is the output? When we gave up the overall reaction of respiration, okay, we said carbohydrate is oxidized in presence of oxygen. Now, till Krebs cycle in my previous class, I never talked about oxygen, okay. Now, ETC is the place where we are going to talk about oxygen. How? Because at the end of ETC, the last step, oxygen is going to get reduced to water molecules. So, that is what is this output we are talking about, and it's going to release ATP also, the water molecule on reduction of oxygen. What is the difference between CLP and SLP? It's nothing but I'm talking about chain level phosphorylation, and SLP is substrate level phosphorylation, okay. I have just given the first letters, I have not given the expansion, it's just the abbreviation. So, in chain level, what we are talking are ETC, there is no direct synthesis of what is that ATP, okay. All the protons which is released from electron transport chain is used to drive off the synthesis in ATP synthase, okay. It's little complicated when you compare to substrate level phosphorylation, which we have been studying in glycolysis, Krebs cycle, where at the substrate level there is formation of ATP. I will not go into the details of it, we have already discussed about it. Then, most important thing to note what is happening in ETC. We put in two different colors, underline, okay. Each coenzyme, what is that coenzyme we are talking about? That is the NADH and FADH. Each coenzyme uses one oxygen atom in ETC. Just keep this in your brain. I'll explain it to you that how it is happening through ETC. Just keep it that each coenzyme utilizes one oxygen atom, okay. Composition of ETC, I just now showed you the chain, right. There are 4 complexes and 2 small electron carriers, okay. That is what ETC is completely made up of. This is sitting inside the, which is the inside the inner membrane of mitochondria. This chain is sitting inside inner membrane. Whatever protons is getting released or gets accumulated in the intermembranal space, that's how, which is going to drive off ATP synthesis. You see here we have magnified this. This is ATP synthase. ATP synthesis takes place in this complex, which is called as complex 5. Then force which is driving off ETC, I said no, like a cookery class. In cooking, what is driving cooking is our natural gas, the gas. So in ETC, what is it that is driving off ETC is our chemiosmotic gradient or proton motive force. You must have studied this in your photosynthesis class when our faculty would have taught you about non-cyclic photophosphorylation, okay. There should have talked about how proton motive force is actually driving off ATP synthesis. The same principle here, okay. So ETC at a stretch. Here I want you to be little attentive, keep an important note on every step as I go through. I am going to take you along with me inside the inner membrane of mitochondria and see how there is going to be electron transfer. I just now told you the pushing away of electrons stepwise. Why is it moving in downward direction? Yes, each step we are going to discuss that. So, for a couple of time, just with 2 slides, imagine yourself that you have gone inside the inner membrane of mitochondria. Along with me, I am going to be the narrator. You are going to imagine this and also note down what is happening in each step. Cool? Okay. So, this chain which I have drawn here, let me use a highlighter, okay, the laser pointer, sorry, not the highlighter. So, this entire thing, this entire chain which I have drawn is inside the inner membrane of mitochondria, okay. So, this outer part is the matrix of mitochondria. You see that folding, right? Let me just draw it. So this is the inner membrane inside the mitochondria, right? So we have been talking about this position where ETC, the chain is here. This is the matrix. This is the intermembranal space. So this is the membranal space. Okay. Yes, kids. First step with full enthusiasm. Let's keep looking at this. It is so beautifully mechanized inside the smallest structure, mitochondria, and within that, inside the inner membrane of mitochondria, this is taking place. How beautifully it is, taken place. Just check out. Yeah. So the NADH, as I said, from glycolysis, Krebs cycle and pyruvate, it's gonna enter. I'm just gonna use the laser pointer. It's gonna enter into complex 1. You have to memorize a little bit here. Okay. Complex 1 is called as NADH-Q reductase. Okay. It's called NADH-Q reductase. It's gonna be our complex 1. It is sitting inside the inner membrane of mitochondria. Our reduced coenzyme from glycolysis, it is in the reduced form, right? It undergoes oxidation by releasing its proton to its prosthetic group. One more thing which you have to keep it in note is the prosthetic group of complex 1. Okay, this entire thing is complex 1. This entire thing is complex 1 is FMN, which I have written it here. You can see it. I have written it here. FMN is flavomononucleotide, which is the prosthetic group of complex 1, which is going to accept the protons given by NADH. It is going to get reduced. Okay, it may look a little complicated to you. As we get time at the end of the class or in the next class, in the NEET slide, we are going to draw this again. I'll tell you the trick to draw it. It's pretty simple, but please keep a note on every step as I'm explaining it. Okay. So this is the reduced form of FMNH2. It's the prosthetic group of complex 1. Now, as the proton has been lost from NADH to FMN, the reduced FMN, okay, transfers the electrons. We were talking about protons then, now it is the electrons to something called as ubiquinone. Ubiquinone is the first small electron carrier. I was saying that, right? The small electron carrier. There are 2 electron carriers. We have talked about ubiquinone here. Ubiquinone takes in the electron and gets reduced to ubiquinol. I have written it here. Ubiquinone to ubiquinol. This oxidized form is ubiquinol and reduced form is ubiquinol. Any problem till now? If you haven't understood, kuch dikkat hai aapko, please put it inside the comment. I'll help you out to, we'll again talk about the steps which you haven't understood. Till now I feel it is clear. Okay. This reduced ubiquinol loses its electron to complex 3. Wait, there is complex 2 here. I talked about complex 1 here. Then I'm going to complex 3. Where is complex 2? We'll talk about complex 2. Okay. But first let's talk about complex 3. The electrons lost by your NADH, it moves through complex 1, that is through the prosthetic group. Then it comes to ubiquinone, the first small electron carrier, and then the electrons move to complex 3, which is actually made of 2 such molecules. Okay. Complex 3 is called as QH2 cytochrome C reductase, or just simply cytochrome C reductase. This is actually made of 2 such cytochromes, that is B and C1. Okay, cytochrome C is made up of 2 things, B and C1. So at first the electrons enter into cytochrome B, which gets reduced back to cytochrome B reduced form. Okay, till now we talked about NADH entering. Now we'll talk about the coenzyme FADH. We saw the step where there is going to be release of FADH2 in Krebs cycle. I'll write it in the side for you. We studied about succinate getting converted to, this is in the Krebs cycle, okay, to fumarate. You remember I had encircled F, right, where this is the place where FADH2 is formed from FAD. The enzyme which was involved here is succinate dehydrogenase, okay. So, the enzyme of Krebs cycle is linked to ETC. This enzyme is called as complex 2. So, the electrons or proton transfer from FADH2 is from complex 2 to ubiquinone to complex 3. Note this difference. NADH is from complex 1. I will put a laser pointer here. NADH is from complex 1, okay, to ubiquinone to cytochrome 3. I am sorry, the complex 3, which is called as cytochrome C reductase. And entry of FADH is, or FAD is from complex 2 to ubiquinone 2 or complex 3, that is cytochrome reductase. Fine. Let's see what's happening with, I told cytochrome C reductase is made up of cytochrome B and C1. We talked about B, we'll talk about C1. So yes kids, क्या हो रहा है यहाँ पर? Cytochrome C1 again electrons cytochrome B से accept कर रहा है. As this is a big chain, I've just split it off. It's not, don't split it in your exam or in your descriptive question. It is going to be continuous itself. Okay. So electrons from B enters C1. Okay, it is going to reduce it to C1 from oxidized state to reduced state. This is this complex 3 itself. Now the electrons are going to get into another small electron carrier. See, I'll talk about the release of protons once we are done with the transfer of electrons. Okay, that's why I'm not talking about it, but this also happens simultaneously. You will get confused. That's why we have been talking about electron transfer only at first. Okay. So cytochrome C is a second small electron carrier. It's not a complex, it's like ubiquinone. So the electrons lost by, everywhere there's 2 electron loss only, but here I have given it emphasizing is showing that there is 2 electrons loss to cytochrome C, which is in oxidized state. It gets reduced at reduced form cytochrome C. So the carrier, electron carrier from complex 3 to complex 4 I have given here is cytochrome C oxidase. Just like complex 3, which is made up with molecules of B and C1, our cytochrome C complex is made up of 2 molecules A and A3. Just a second. Use the eraser and make it much more neat for you. Okay. A and A3. Okay, that is why I have given A and A3. We split up B and C1, but here we are not going to split up A and A3. So the electrons which is lost by the reduced form of cytochrome C, it enters cytochrome A, A3. Okay, this complex is called as oxidase because that's going to be the last complex. Okay, as soon as it receives the electrons, it's going to give it off to oxygen. So which is the complex of ETC from where electrons is been lost to oxygen? The last complex, if anybody asks you, is complex 4, named as cytochrome C oxidase. The other enzymes was been named as reductase. This is the only complex which will end up with saying oxidase. Okay. Now our beautiful Mahashay, oxygen, to form 2 molecules of water, it's going to require 4 electrons and 4 protons. 4 protons actually comes from the inner, the intermembranal space. Okay, it's going to come up with the space which has been dividing outer and inner membrane. So oxygen with 4 protons, 4 electrons forms 2 molecules of water. In your textbook it would have been given half molecule of oxygen in presence of 2 electrons and 2 protons forms a molecule of water. This is in your textbook. Both are correct, okay, any one you can memorize it, both are correct. To form 2 molecules it requires 4 protons and 4 electrons. Fine. So we have completed it, okay. The electrons at the end which is being accepted is by oxygen, okay. Now let's go to the previous slide. I'll tell you where and all protons are getting released, because as I told, ATP synthase or complex 5 is waiting for the protons. So here as the electron gets transferred, there are specific junctions where protons are getting released. This is going to drive off ATP synthesis. I hope you are understanding, okay, you are understanding. If any problem is there, put on your comments, okay. So from complex 1 to ubiquinone, I'll use the laser pointer, complex 1 to ubiquinone, there is release of 4 protons, okay. That is from complex 1, I'll write it here, to ubiquinone, there is 1 proton release. From complex 3 to cytochrome C, the another carrier, there is going to be release of 4 protons. That is from complex 3 to cytochrome C, there is release of 4 proton, another junction. The third junction where proton is going to release is from complex 4 to oxygen. I'll write it as OXY, okay. The release of another 4 proton. Complex 1 to ubiquinone, complex 3 to cytochrome C, complex 4 to oxygen. Wherever these are the junction where there is electron transfer, protons are getting released. 4 proton. Don't keep a इतना दिक्कत और ज़्यादा कष्ट मत लीजिए कि कितने numbers of protons थे भाई, कितने थे. Just know that protons were released at this junction. So we are done with studying the electron transport chain. We have seen how electrons beautifully get down stepwise to oxygen. Oxygen is getting reduced to form, what is it? Water. So that is why as oxygen is seen in the step of aerobic respiration, ETC can also be called as aerobic part of aerobic respiration. Finally in ETC, हमने बोला overall ETC हम पढ़ेंगे, right? Any process, anything which is taking in a cell, obviously it's going to have a purpose. It is going to become valuable to the cell. How is it? Now, one molecule of NADH, the protons released by one molecule of NADH, just know this, okay, is going to help us out to produce out 3 molecules of ATP. How is ATP is getting generated? I'll talk to you in the next slides. One molecule of FADH2 is going to help us out in producing 2 molecules of ATP. You have to memorize this, okay. 1 molecule of NADH to 3 molecules of ATP, 1 molecule of FADH to 2 molecules of ATP. Why? Because in the next class when we are going to talk about the balance sheet, that is throughout the aerobic respiration with all the steps, at the end we are going to crash with ATP synthesis. How many ATP molecules were produced? To know that you have to memorize this, okay. One more thing I had told you in the previous slide when we talked about to know more ETC. This is another one, to know more ETC, okay. So I said each coenzyme requires 1 atom of oxygen, okay. That's it I'm going to tell you. The next point here is for every coenzyme which is formed till now, that is till Krebs cycle, it's going to total off to 10 NADH. How it is 10 NADH is? Just look at my fingers. 2 from glycolysis, okay students, 2 from link reaction, it became 4, okay, and 6 from where? Krebs cycle. We talked 3 from 1 pyruvate, it will become 6 from 2 pyruvate, 2 from glycolysis, 2 from link reaction, 6 from where? Krebs cycle. So that's how it becomes 10 NADH molecule and 2 from Krebs cycle. This side we are talking about Krebs cycle, This side we are talking about glycolysis and link reaction. So this side 2 molecules of FADH. I think this type of monoacting will help you out remembering how many NADH, how many FADH was out. I hope I'll be extremely happy if this monoacting has helped you out in understanding how many molecules where and what is getting released. Okay. So this completely becomes 12 molecules of coenzyme, right? 12 molecules of coenzyme using only 6 molecule of oxygen, isn't it 12 molecule? Question yourself, think about this, we'll talk in the next class. How is it 6 molecules? So how is the path of NAD and FAD's protons released? I just now talked, I've just summarized here. So the protons released from, see, keep a note on this also, many unique questions come up with this concept, okay, in your NEET, okay. So the electrons lost from NAD travels from complex 1 to ubiquinol to complex 3 and goes to complex 4. There is no involvement of complex 2 here. So FAD is from complex 2 to 3 to 4 through ubiquinol it gets in, okay. No complex 2 here because FAD is involved in complex 2. So 1 to 3 to 4 NAD, 2 to 3 to 4 is FAD, the proton movement. Know this, many questions have been asked, unique typical questions, you know. So at that time if you are like where is this coming from, what they are talking, so just know this concept, okay, then you will understand. Previous class contest question, reducing agent or reduced agent of glycolysis is dash in anaerobic respiration. My friends, it is oxidized. I see that nobody has answered this question. I think you haven't understood what is a reducing agent is. Reducing agent is nothing but our reducing cofactors, that's nothing but coenzymes NADH and FADH. So as we are talking about glycolysis, we will only talk about NADH, okay. Okay. So NADH was utilized to reduce pyruvate. We were talking in fermentation, right? In fermentation pyruvate got reduced, okay, by NADH. So NADH of glycolysis is oxidized. Now when it is utilized for reduction, it will get oxidized. So it is oxidized in anaerobic respiration, okay. After reducing pyruvate, NAD comes out, right, that means it's got oxidized. Simple, no? It's got oxidized in anaerobic respiration. I hope you have understood the concept behind it. We will go to our contest time, our contest question. If you have kept a keen note on everything and whatever we talked till now, okay, the question will be raised is, in ETC the final acceptor of electrons is cytochrome C oxidase, water, oxygen, all of the above. It is a pretty simple question, I would say, an extremely straightforward question. Okay, answer this correctly. Let's see how many of them come up. Okay, come up with the correct answer and take the atom session given by us. I was talking so long that ATP synthesized in ATP synthase, ATP synthesized in ATP synthase. Now this is the right time. With this slide we are going to talk about oxidative phosphorylation, okay. See, this complex 5 in your textbook actually says ATP synthase, but it is not involved in ETC, it is something separate. So all these protons which got released out is going to enter our beautiful ATP synthase. I have put down here the points to help you out, okay. Keep a keen note on all these points as we keep discussing. Complex 5 is ATP synthase, kids. My dear students, it happens inside mitochondria. So mitochondrial ATP synthase is very much similar to that of chloroplast. I just now told you that we talked about this in a photosynthesis chapter in detail. It's going to be the same way how it's going to work out. Okay, the components of ATP synthase, see this diagram I've put, ignore all the extra labelings which are there. So the head piece, you see this, the head piece is the F1 component, that's why it is called as peripheral membrane complex, protein complex. Peripheral because periphery means something at the edge, something at the peripheral area. So if this is the membrane, your head is going to sit like this, okay. So head piece of ATP synthase is called F1, that's why it's called as peripheral mitochondrial protein complex or membranal protein complex. F0, you see this long thing which is inside the inner membrane, across the inner membrane, that is called as F0 component. It is called as integral membrane protein complex. Integral membrane is because, see, just think, if this is the membrane, okay, here is going to be the head piece, as I told. Across this membrane, your, you know, F0 is going to sit like this. I hope you are able to picturize this, okay. So F0 and F1, F0 is across the membrane, it is the one which helps for the passage of protons. You see the protons here in the intermembranal space, all these protons is going to get in through F0, and F1 is the head piece where from ADP and inorganic phosphate ATP is formed, okay. This is the area where ATP is formed. One more thing to note on, 2 protons are involved in production of 1 ATP. Now let's give justice to this name, oxidative phosphorylation. Why oxidative? Because on oxidation of a reduced coenzymes, let's make it as FADH2 NADH plus. Oxidation is oxidation of macromolecules to release energy. When there is oxidation in terms of metabolism, we are supposed to call it catabolic because the energy is getting released. Photosynthesis is called anabolic because we use the energy to form something, but here whatever is formed is undergoing breaking down. It's broken down or oxidation, that's why it should be called as catabolic. But my friends, it is called as amphibolic because both catabolism and anabolism is going to take place in respiration. That I'll tell you. So before that, do you think respiration is merely catabolic? I just now told no. Is carbohydrate the only respiratory substrate? Do you think? Is carbohydrate the only respiratory substrate? There are many conditions where the plant undergoes severe condition where it can't photosynthesize, or it has to rely on another components for respiration to occur. We had discussed that in the as we started off the class, that is respiratory substrates can be carbohydrates, fats, proteins, organic acids also, okay. So it is not only the carbohydrate which is our respiratory substrate. Types of metabolism, we studied 2 types, anabolic and catabolic. I am not going to write their definition. Catabolic is breaking down, anabolic is forming something. Amphibolic pathway, we are going to draw it now, okay. This is the, you know, I'll use the laser pointer here. This is the entire route what a carbohydrate enters till our Krebs cycle. We have just made it shortened off. If you see it in a textbook also, this entire scheme is given, okay. Carbohydrate through glycolysis converts to glucose phosphate, then to fructose phosphate, then many steps takes place to DAP and PGAL, okay. We talked about it, no, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This is glyceraldehyde 3-phosphate. This will get converted into pyruvate. This is the entire step till pyruvate to glycolysis, that is glycolysis. Pyruvate to acetyl-CoA is our link reaction or pyruvate oxidation. Acetyl-CoA gets inside Krebs cycle, okay. There's so many reduced coenzymes getting out. So with this Krebs cycle, carbon dioxide gets out, the water gets out. So this is the entire shortened summarized scheme of what is the fate of carbohydrate through glycolysis, pyruvate oxidation, then Krebs cycle. I told there can be proteins also as our respiratory substrate and also fats as a respiratory substrate. When carbohydrate amount is less or the plant has to depend on others respiratory substrate, proteins is broken down into amino acids in the presence of protease enzyme. Fats is broken down into fatty acid and glycerol. Okay. Now this is getting broken down, that means there is catabolism happening to proteins and fats. This fatty acid which is formed, it enters respiration into acetyl-CoA. You must have studied in biomolecules chapter how fatty acid can get converted into acetyl-CoA. So as it gets converted to acetyl-CoA, it will simply enter into Krebs cycle. Glycerol, it will get converted into our DAP, dihydroxyacetone phosphate. Okay, please note down to the arrows. Okay, and then it gets inside glycolysis, it undergoes oxidation, and then it goes inside your Krebs cycle. Amino acid, it has various steps to get in actually. Can get in by forming pyruvate also into glycolysis, or it can get inside Krebs cycle also. How it is actually is, you must have studied about transamination in mineral nutrition chapter. There alpha ketoglutarate acid in presence of amino group, that's NH4, it gets reduced to form many amino acids. So this amino acid will break down to itself to form alpha ketoglutarate acid, and then it will get inside Krebs cycle. You know alpha ketoglutarate acid is formed in Krebs cycle. Okay. So this is the various ways by which the other respiratory substrate gets inside respiration and oxidize themselves to give in energy. See, the number of ATP molecules which is going to be spreaded out or given out by these respiratory substrates are going to be different when you see it in terms of carbohydrate. It's not necessary to know now, okay, but they are also helping us to give out ATP. At the end with amphibolic, I told respiration is both. What is this? It is catabolic and anabolic. Okay. How it is? It is actually is this fats which got burned into fatty acids, okay, and glycerol, it is going to enter into acetyl-CoA and PGAL, we just now discussed. So as it got burned out, it underwent catabolism. There are certain cases or there are certain condition in the plants where it says you used it for oxidation, now I need it for some other purpose. I need fats and fatty acid and glycerol for some other purpose, because these are all building molecules, they will be involved in other metabolic activities also. Now in a reverse way, this has to get anabolized, in the sense all this acetyl-CoA and PGL which was formed on breakdown, now it will undergo anabolic pathway to form back the fats through fatty acid and glycerol. So that is why breaking down through catabolism it enters respiration. Now when it has to get formed back, anabolism takes place. Understood? Now both the things are happening, and that's why respiration is called amphibolic pathway. That is the same condition with amino acid also. It is formed by transamination, I just now told you, with alpha ketoglutarate acid. Amino acids are formed. It undergoes deamination through catabolism. That is, when amino acid has to break down, it undergoes deamination. When it has to form back, it undergoes transamination. Okay, keep a note on all this. Cat I have given is catabolism. Reverse ana is actually reverse anabolism is taking place. Now we have discussed that respiration is amphibolic. When anybody asks you respiration is catabolic or anabolic, you should say amphibolic. Amphi means staying in both the places. That's why we say amphibians also. Well, that is why respiration is called as amphibolic. Yes, my friends, we are at the end of the class. We beautifully discussed ETC. We told overall ETC what are the things which we are going to see in ETC. A step more to know ETC, because these step more to know ETC is going to help you out to solve out the tough questions, or to answer the tough questions will come in your entrance exam. Now at the end we told respiration is amphibolic pathway. What did we do? Let's summarize. We studied ETS, we studied oxidative phosphorylation by studying ATP synthase. We studied overall ETS or ETC or and amphibolic pathway. We're done with the class. So when you come to the next class, we are going to summarize all the steps and tell that how many molecules of ATP is getting released, that's through balance sheet. And we are also going to talk about something very interesting, that is RQ factor, okay, the respiratory quotient for different respiratory substrates, okay. So before you come into my next class, I want you to brush up glycolysis, Krebs cycle, link reaction and also ETC. So it will be helpful for us to summarize ATP synthesis, how many molecules are gonna get out, okay. Thank you for being tuned with us. All the very best. And for our passport students, please answer 10 CPP questions. It's all made on NEET perspective. Answer those. Any problems, get back to us through comments. Okay, we'll be happy to see you in the next class. Have a great day ahead. All the best.